Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive. Today we're looking at how AI is stepping way beyond just making pictures. It's actually designing tiny molecular tools that could, well, really shake up medicine.
Yeah, it's pretty exciting stuff.
So let's unpack this, the headline here, the really key takeaway. Researchers use generative AI to build this incredibly small protein. We're talking just 82 amino acids.
Tiny.
Right. And this tiny thing acts like a booster for precise gene editing. Prime editing bumping up the efficiency by, get this, up to 18.8 times.
And that boost, that 18.8-fold increase, it's not just a number. It hits right at the core limitation of prime editing, or PE.
Okay, so remind us quickly. PE is the super precise search and replace tool for DNA.
Exactly. It's the one we really want for therapies because it avoids those risky double-strand breaks you get with older methods like CRISPR-Cas9. Much cleaner.
But the cell doesn't just sit back and let us tinker with its DNA, does it? There's a defense mechanism.
Right. There's always a defense mechanism. In this case, it's the Mismatch Repair system, MMR. Think of it like the cell's quality control.
Yep.
When prime editing makes that specific change you want, it creates this temporary structure in the DNA that looks, well, wrong to the cell, like a typo.
A mismatch.
Precisely. So the MMR system, specifically a protein complex involving MLH1, rushes in, sees the typo, and corrects it back to the original sequence. It basically undoes your edit.
Ah, the undo button strikes again. And people have tried to block this MMR system before, right? But there were issues.
Yeah, the main approach involved using these big, bulky molecules, like dominant negative versions of MLH1 itself, to try and interfere.
And big and bulky is a problem for delivery.
Exactly. It's the problem for delivery in gene therapy. You need to package your editor, and potentially an inhibitor, into these tiny delivery vehicles like AAV viruses or lipid nanoparticles, LNPs.
Which have really strict size limits.
Very strict. So you just couldn't realistically fit both the prime editor and a massive MMR inhibitor into one package. The field needed something much, much smaller, a sort of molecular saboteur that could slip in easily.
Enter the AI. Okay, so figure one. How did they actually design this tiny saboteur, this MLH1 small binder, or MLH1SB?
Well, they started with a generative AI model called RFdiffusion. It basically dreamt up thousands and thousands of potential small protein structures.
It's generated possibilities.
Right. But just designing a stable protein isn't enough. You need one that actually does the job, in this case, physically block MLH1 from doing its job.
So how do you check that without testing thousands in the lab? That sounds expensive.
This is the clever part. They used a second AI, a new structure prediction model, AlphaFold 3. In a really novel way, they set up a competition.
A competition between proteins.
Sort of. They used AlphaFold 3 to predict whether their little AI design binder could actually push aside MLH1's natural binding partner, PMS2, and bind to MLH1 instead. They specifically targeted 3 spots on MLH1.
So the first AI designs the key, and the second AI tests if the key actually fits the lock and can elbow the other key out of the way.
Exactly. It's like a virtual stress test. This let them filter out tons of designs that looked good on paper but wouldn't actually compete effectively in the cell. Saved a huge amount of time and resources.
Okay, so they've got this winner, a tiny 82 amino acid binder that works in the simulation. Now, the next hurdle, packaging. You've got the editor, you've got this new booster. How do you make sure they both get where they need to go together?
Well, thinking about it, if you package them separately, you risk one arriving without the other, or maybe not in the right amounts. What's the most foolproof way to ensure they work together?
Maybe. To stick them together, like fuse them into one molecule.
Precisely. Combine them. Why introduce delivery uncertainty? If you fuse the binder directly to the editor, they have to arrive together in the perfect 1-to-1 ratio.
And looking at figure 4, that's exactly what they did. They took their best small binder, the MLH1-SB, and literally stitched its genetic code into the code for the prime editor. They used a 2A self-cleaving peptide.
Yeah, that just means the two proteins are made as one piece initially, but then they automatically snip apart inside the cell to function independently while still ensuring they were delivered together. They called this new integrated system PE7SB2.
Clever. And the result?
Really impressive. In their lab tests using HeLa cells, this all-in-one PE7SB2 system showed that massive 18.8-fold increase in editing efficiency compared to the standard PMAX system.
Wow.
And even compared to the already improved PE7 editor without the binder, it still gave a solid 2.5-fold boost. So fusing the tiny inhibitor directly onto the editor worked incredibly well.
It's like the editor now carries its own shield against that MMR undo button.
That's a good way to put it.
What's really innovative here, it seems, is the shift in focus. A lot of work has gone into making the editor itself better, tuning the guide RNA, evolving the reverse transcriptase.
Right, improving the search and replace parts.
But this tackles the other side of the equation, the cell's resistance, the repair pathway. And it does it with this compact modular add-on.
Exactly. And crucially, that compactness, just 82 amino acids, is the game changer for delivery. It's small enough to fit comfortably alongside the editor within those tight AAV or LNP limits.
So this overcomes a major bottleneck.
A major practical bottleneck for actually using prime editing as a therapy. This could pave the way for a kind of standardized, highly efficient editing package, the editor plus the MLH1 binder, making precise gene therapies much more feasible.
Okay, let's quickly recap the main points from this deep dive. First, AI was used to rapidly design a very small protein, MLH1SB.
Yep, the binder.
Second, this binder works by specifically getting in the way of the mismatch repair protein, MLH1, stopping it from reversing the edit.
Blocks the undo.
And third, fusing this binder directly to the prime editor created the PE7SB2 platform, which boosted editing efficiency by up to 18.8-fold in their tests.
Huge improvement.
Now, there's one key limitation mentioned. This big efficiency gain mainly applies to smaller edits, right?
That's correct. It works well for substitutions, small insertions, small deletions, things under about 12 base pairs. If you're trying to insert much larger DNA sequences, say over 23 base pairs, inhibiting MLH1 doesn't seem to help much based on the study. That remains a challenge.
Still, pretty remarkable progress for smaller edits.
Absolutely. And it leaves you wondering, if AI can design these highly specific tools to, well, jam fundamental cellular machines like MMR this quickly, what other biological roadblocks, maybe in diagnostics or other therapies, are suddenly looking solvable, not in years, but maybe, much, much faster?
A powerful thought to end on. That's all for this deep dive.